Hi there. In this video, I talk about the central dogma, but go beyond to talk about some of the things that are hidden within the expression of genes that the central dogma often bypasses. So I'll start here. Here is the central dogma sort of simplified, and this was something that Francis Crick first proposed in the late 1950s, and almost immediately after he did, he kind of regretted it because it oversimplifies the process. So essentially it says that DNA, which can be replicated, is transcribed into RNA, and the RNA then is translated on ribosomes to produce protein. So there's this movement of genetic information from DNA into protein. However, in an evolutionary sense, as we've already discussed, RNA was one of the first molecules in biological organisms, and then RNA plus peptides, and then eventually RNA plus proteins, and DNA was a later arrival, a few 100 million years later. So the first biological processes started this way. So evolutionarily, this central dogma is not the way the evolutionary process proceeded. So although DNA is a much more stable molecule for maintaining genetic identity of genes and whole genomes, RNA originally was the genetic material as well as some of the enzymatic materials also, and some of those are still around today in modern cells. The ribosome being one of the largest examples of that, being enzymatic as well as structural and functional. So eventually there were proteins formed from this translation process, but at that time there were RNA molecules that preserved the genetic material, and there were RNA molecules, several different ones, that together performed translation to make proteins. So that's the second part of the flow of genetic information that evolved. And then finally, a relative of RNA, that is DNA, was used for the genetic material, and it was much more stable and could form long-range double-stranded molecules that would protect the genetic material and would provide a check on any mutations that happen, and RNA just is not capable of either one of those. So now we finally have the flow of genetic information that we see today. So then we have the central dogma, and now I want to talk about this in a revised way. So we do have DNA replication, which we've talked about a little bit, and transcription, which we've also talked about, and then finally translation, which we've also discussed in some of the previous videos. So if we pull this apart piece by piece, here's the DNA, and it's now almost exclusively just the genetic material for most organisms. There are some viruses that still use RNA as their genetic material. So DNA is replicated by DNA polymerase, which still uses RNA primers to start the reaction. And it's related to RNA polymerase. It's also more distantly related to reverse transcriptases and also RNA-dependent RNA polymerases. Next, if we move on to transcription, in bacteria, there's really only one RNA polymerase, but then there are different subunits that affect whether it transcribes ribosomal RNA, tRNA, or mRNA. But in this case, I'm talking about eukaryotes, where you have three different polymerases. Again, they are closely related. The central parts of them are the same. It just is a difference in subunits that allows them to either transcribe ribosomal RNA, tRNAs, or mRNAs. So in this case, we have POL1, which only transcribes ribosomal RNA, and primarily the large ribosomal RNA including the small subunit, large subunit, and the 5.8S genes. And recall that these RNAs act as RNAs. They are never translated. They act to translate proteins, but they themselves never are translated. So they act only as RNA molecules. So ribosomal proteins are added to those to hold them in their catalytic conformation or their functional conformation. And you have ribosomes assembled then from this ribosomal RNA. Next, RNA polymerase III in eukaryotes transcribes tRNAs as well as some small RNAs. So primarily tRNAs, but also some small controlling RNAs. Next, the amino acids are added by aminoacyl tRNA synthetases, a protein that adds the amino acids onto the acceptor arm of the tRNAs. So now they're charged with their attendant amino acids, ready for translation. And as with ribosomal RNA, these are never translated, right? They take part in the translation process, but they act as RNA molecules. They are never translated into proteins. And finally, we have POL2, RNA polymerase II in eukaryotes. It's polymerase in bacteria with different subunits. And this produces mRNA. Now, mRNA is the coding part, so this encodes for proteins and otherwise isn't functional for anything else. And next is processing of the mRNA. So you have addition of a 7-methylguanosine cap, and then introns are spliced out of that mRNA, if there are introns. And then a poly-A tail is also added to the end. And then finally, it's shipped out of the nucleus through the nuclear pores. And again, in bacteria, the process is simpler, but it follows the same path. And then finally, these processes come together in translation. So the ribosomes then grab onto the mRNA. tRNAs are shuttled into the ribosome and drop their amino acids in the proper sequence according to the DNA or the RNA sequence of that mRNA to produce those proteins necessary for the organism to survive. So what I've outlined in red here then is essentially the central dogma of molecular biology, or the flow of information from that central dogma, and it ignores or sort of puts in the background the ribosomal RNA and tRNA that are accomplishing that second step of translation in this central dogma, in this pathway of the flow of information. So we can discuss each part here. This red is essentially the flow of information, and the blue, outlined in blue here, is the machinery that accomplishes that process, especially the last part of that process, which is translation of the message, the mRNA, into an amino acid sequence or a protein. And all of the processes here that I've mentioned are dependent on these proteins, which are responsible for transcription, translation, and replication. So there's a whole feedback of all of these components to produce a living organism or a living system. So here it is full circle, where each one of these proteins that's produced over here is really feeding back into the system. So you need DNA polymerase and other proteins to replicate the DNA. You need RNA polymerase and transcription factors to accomplish transcription, and that's whether it's a bacterium or a eukaryote. And ribosomal proteins, amino-acyl tRNA synthetases, and other proteins in spliceosomes and things like that are also used in the system. Most of this evolved, or at least the rudimentary parts evolved some 4 billion years ago, in a much simpler form. But it still evolved in the same way, so that you had ribosomes consisting of ribosomal RNA, maybe some peptides, and then you had a system for reproduction of the RNA, probably an RNA-dependent RNA polymerase. And then the ribosomes, or the ribosomal RNA with the tRNAs, which were also around at the time, accomplished producing peptides at first, which are amino acid sequences less than about 50 amino acids, and then later the proteins became established as much longer polypeptide chains of greater than 50 amino acids. But the basic parts of this, that are still around today, evolved some 4 billion years ago, and are still going today, although little bits and pieces are being added to them all the time. For example, the amino-acyl tRNA synthetases probably were originally RNA enzymes. Now they're protein enzymes. The processing of introns is currently done by either protein RNA complexes or RNA alone. And so that's a process that's very ancient also. And ribosomes now have many proteins attached to them, primarily to keep the ribosome, the RNA, in its catalytic and structural form. Also, very early on, there were fewer than 20 amino acids that were utilized, and probably the first genetic code only encoded about 4 amino acids, and then started expanding from there to the current number of 21 if you include selenocysteine. Here is the process in a little greater detail. Here's the DNA, and it becomes replicated with DNA polymerase, and some other proteins are involved as well. Transcription by pol 3 to make the tRNAs and small RNAs, and there's actually a pol 4 and 5, mainly in plants, that produce other RNAs, small RNAs. Pol 1 produces ribosomal RNA, the main transcript for ribosomal RNA, including the small subunit, large subunit, and the 5.8S gene. And then here's what produces the mRNA. And at first, if it has introns, it's heteronuclear RNA, and then is processed to become the mature mRNA. Eventually, the tRNAs and the ribosomal RNAs come together as the activated translation machinery for reading the code on the mRNA to produce the proteins. And there are a few other RNAs also, mainly small RNAs. Some of these are parts of introns that are freed up during processing of the larger precursor mRNA. And some of those become small nuclear RNAs that control or help splicing of the heteronuclear RNA. Others become microRNAs to control the expression, the when and where the mRNA is translated, or if it is translated. Some of those become small nuclear organizing RNAs, which process ribosomal RNA, and again, these are enzymatic RNAs. And then there are these silencing RNAs in plants that are transcribed from pol 3, using pol 3, that also control the expression of that mRNA when or if it is ever translated. So again, these lines in blue, here, and here, that's the original central dogma that was proposed by Crick back in the late 1950s. But we've discovered a lot more, and even at that time, he realized that more was involved. Ribosomal RNA had been discovered because it was one of the most prevalent RNAs in a cell. tRNAs had been discovered, but they didn't know exactly what they did at that time, and that was only discovered a few years later in the 1960s. And introns were discovered and studied in the 1970s, and it wasn't until about the 80s or 90s that these small RNAs started to be studied as something real rather than just degradation products. So we now know that there are a lot of small RNAs that perform many functions in modern cells, and they were probably there a lot further back in time. As I indicated in one of the first slides, that RNA was one of the first molecules in used by living organisms. And it was only later that DNA was used, and RNA performed many, many functions and still does. For instance, ATP is an RNA molecule. NAD and NADH are RNA molecules, and there are ribozymes, the hammerhead ribozyme, group 1, group 2 introns, and spliceosomal introns, and the spliceosomes themselves are composed of RNA and protein. So many, many functions for RNAs in cells. So RNAs can be mRNAs, carry messages, and that's what most people think of when they think of mRNA. But since I studied ribosomal RNA in my PhD work and ever since, you realize that RNAs can be structural in the form of ribosomal RNAs and tRNAs and others. They can be actually structural elements. They can also be enzymatic. So ribosomal RNA is enzymatic. Some introns are enzymatic, and spliceosomes certainly are, as well as many other ribozymes that are in cells. And then they can also be controlling. So those small RNAs and microRNAs, those can control expression in a variety of ways, actually, to determine when or if a particular mRNA or a batch of RNAs are ever translated. And it may be that the RNAs can hold on to those mRNAs for a while, and then upon a signal, release them to the ribosomes for rapid translation into proteins. So if you just look at the amount of RNA in each class within a cell, it breaks down approximately this way. So by mass, so by weight essentially of the RNA, 80% of the RNA in a cell is ribosomal RNA, and that's mainly almost all in ribosomes. So you need a lot of ribosomes in a cell, 80%. That's about 10 million molecules, although depending on the cell, it can vary between about a million or 2 million up to 10 million, sometimes even more. In bacteria, that would be lower, probably in the tens of thousands of ribosomal RNAs. But there's only 4 to 6 different genes, so the gene diversity is low. If you look at tRNAs plus other small RNAs, package that all together, that makes up about 15% of the total RNA, about 20 million molecules. Because these are small molecules and have a small mass, therefore, that's why you have 20 million of them make up only 15% of the total. And there are 20 to 30, sometimes up to 50 or 60 different types of tRNAs, and then dozens or hundreds of small RNAs. So that gene diversity is also quite low. So then you finally get to this, the mRNAs, makes up about 5% of the total RNA by mass, and only about 350,000 mRNAs in a eukaryote, probably in the thousands or tens of thousands in a bacterium. But there are between 20 and 30,000 of those, and if you look at bacteria, there are anywhere from several hundred and then up to eukaryotes, maybe 50,000 or 60,000, but generally around 20 to 30,000 different genes. So the gene diversity is very high, and that's where you get the protein diversity, so that's where you get the general overall diversity of a cell. So then about 95% of the RNA in a cell is involved in the protein producing processes, as well as somewhat in expression of those mRNAs. But 5% is in the mRNA, and that's where all of the, or most of the diversity in a cell comes from. And that's usually referred to as the complexity of a cell. So the RNA complexity and the protein complexity is determined by that 5% of the RNA that is in a cell, and it's 95% is the machinery that produces the proteins from those mRNA molecules. So that's looking at the central dogma in a more detailed way, and trying to consider all of the molecules that are involved in the flow of information from DNA to proteins. So that was first put forth again by Francis Crick in the late 50s, but we know a lot more about what is actually going on in there, and I think it helps to look at what molecules, especially ribosomal RNAs and tRNAs, and understand how important those are in the whole system of translating or expressing those genes into proteins. Okay, so that's all I wanted to say about that in this video, and I will talk to you later.